The name of the paper I read was Mapping the Cancer Genome by Francis S. Collins and Anna D. Barker. Renato Del Becco, a Nobel Prize winner, once said, "If we wish to learn about cancer, we must now concentrate on the cellular genome." Him, along with other scientists, thought that by sequencing the entire human genome, it would allow us to understand the behavior of cancer. If we have an entire sequence of the nucleotides in the DNA of the human genome, scientists could pinpoint certain genes and be able to tell what their functions are. With this information, we could then find how these genes are associated with cancer. This led to the creation of the Human Genome Project. The Human Genome Project was an international collaborative research program whose goal was the complete mapping and examining of all the genes of human beings. They found that we have roughly around 20,500 genes with specific locations in the genome that can be pinpointed and identified. One of the project's intentions was to find a way to compare DNA base pairs in the genome of normal acting cells to those of cancerous cells. When studying cancer, we must focus our attention to the cellular genome. This is because all cancers arise as a result of changes that have occurred in the DNA sequences of the genomes of cancer cells. It is damage to the genes or mutations that cause this. This alteration in the genome causes a change in the function of the genes. The Cancer Genome Atlas was launched after the Human Genome Project had generated a comprehensive map of the main genomic changes in 33 types of cancer. There was a great sense of urgency in undergoing this research because of how detrimental the disease has been within our country, killing at least 1,500 Americans daily. We would like to find new ways of identifying and attacking specific targets within cancer cells. The goal of the TCGA is to eventually be able to identify all the genetic alterations that are associated with all types of cancer. As I already stated, cancer is caused by mutations in certain genes. The damage that causes these mutations can be from radiation exposure or toxins called carcinogens. Some mutations might disable genes that normally protect against abnormal cell behavior, while others increase the activity of disruptive genes. And here's a diagram that shows how a proto-oncogene turns into an oncogene, which then results in a cancerous cell. A proto-oncogene is a normal gene that could become an oncogene due to mutations or increased expression. There are many pathways that lead to cancer. As shown by this diagram, these are known pathways just for prostate cancer. No matter what is the cause for certain mutations, the result of cancer is always uncontrollable cell replication or growth. If malignancy occurs, the cancer has the ability to invade other parts of the body. There are 6 characteristics of tumor cells. One is growth signaling on their own. This means that cancer cells generate their own growth cues. Another characteristic is not being sensitive to anti-growth signals. This means that cancer cells become deaf to anti-growth cues from surrounding tissues. Another characteristic is avoiding cell suicide. This means that the mechanisms that should trigger or carry out a self-destruct program is damaged or disabled. Another characteristic is potential for unlimited replication. Cancer cells evade limits of the number of times a normal cell can divide. And another characteristic is continuous growth of blood vessels. This is where tumors emit signals promoting the development of new blood vessels to deliver oxygen and nutrients. Invasiveness and motility is another characteristic. This is where cancer cells defy multiple signals and forces that hold a cell in place and prevent them from traveling to other tissues. Here is a simplified diagram provided by the National Cancer Institute that shows how uncontrolled cell proliferation occurs. The red shapes inside of the cells represent mutations. As you can see, it takes several mutations for the cells to become cancerous. These mutations can be triggered by many different things. For the past 20 years, many research groups have used different techniques in order to find out which mutations cause abnormal cell behavior. So far, about 350 genes relating to cancer have been found, and a database called Catalog of Somatic Mutations in Cancer, or COSMIC, holds a record of these gene changes. However, this is most likely not the complete list, since further research is being performed today. So, a major question is, what is the purpose of identifying genetic changes in cancer cells? And the answer to this question is that identifying specific genetic changes in cancer cells can point to better ways to diagnose, treat, and prevent the disease. The Human Genome Project created a reference sequence of 3 billion DNA base pairs in the human genome. This is of normal human tissue. The TCGA plans to compare this normal DNA sequence to those of cancerous cells. This would allow for the identification of genetic alterations that cause cells to become cancerous. The Human Genome Project has also led to the creation of many new technologies that allow for sequencing and analysis of genomes. In 1990, the cost of sequencing a single nucleotide base was at least $10. Due to advances in technology, the cost is now less than a penny per base. New methods of sequencing are still being developed today, and the cost of performing such methods is expected to become even lower. The BRAF gene was discovered in 2001, and a mutation in the gene was found in 70% of melanoma cases. With this information, drugs were created to stop the function of BRAF. First, the drugs and therapies were created and tested on mice. Just 5 years later, the most favorable of these therapies are now being tested in clinical trials. Other research groups have pinpointed genetic mutations linked to certain types of breast cancer, colon cancer, leukemia, lymphoma, and additional cancers. This has helped with the creation and use of some of the latest treatment options. All of this research also led to the creation of many effective drugs. Gleevec inhibits an enzyme produced by a mutant version of 2 genes that cause leukemia. Herceptin targets a cellular signal against breast cancers with an abnormal multiplication of receptor protein. The drugs Iressa and Tarceva are effective against lung cancer, as well as Avastin for lung, colon, and other cancers. A difficulty for researchers is to distinguish whether or not a mutation that is found is useful. Studies have found mutations that don't overlap in tumor samples of the same cancer type. Studies have also found dissimilarity in genetic mutations found in tumor samples of the same types of cancer. This means that many different combinations of mutations can cause cancer cells to form. The work that goes into finding valuable information about cancer is very complex, and there are over 100 different types of cancer. Obviously, this makes the goals of the TCGA very hard to achieve, and their research is expected to take many years in order to find all of the mutations associated with every type of cancer. The TCGA has begun a project that intends to develop and test for ways of mapping out all of the abnormalities associated with cancer. The National Cancer Institute and the National Human Genome Research Institute put together teams of scientists and researchers to participate in the project. In 3 years, both institutions plan on putting $100 million into assembling an atlas of genomic changes of 3 types of cancers: glioblastomas of the brain, lung cancer, and ovarian cancer. They have goals of scaling up to a larger amount of cancer types if this project is successful. Cancer genome characterization centers, genome sequencing centers, and a data coordinating center are also collaborating and exchanging data to help out with the project. They will use their technology to take a look at gene activity in tumors and try to find out what changes contribute to the development and spread of cancer. There are several genomic changes that will be studied by cancer genome characterization centers to see how they contribute to the development of cancer. Chromosomal rearrangement is a type of chromosome abnormality involving a change in the structure of the native chromosome. Such changes may involve several different classes of events, like deletions, duplications, inversions, and translocations. Copy number variation is a relatively new field in genomics, and it is when sections of the genome are repeated and a number of repeats in the genome varies between individuals in the human population. Epigenetic change is a regular and natural occurrence, but can also be influenced by several factors, including age, the environment or lifestyle, and disease state. The DNA sequencing and genome characterization groups will experience a large amount of complexity when they examine DNA samples of tumor cells. This is because once cells become cancerous, their mutation rates also increase. This makes them more complicated than examining DNA of normal cells. These mutant cells are also prone to more repair mechanism failures and other factors that affect their ability to have self-control. All of this complexity makes it hard for researchers to distinguish which parts of the tumor genome are significant. However, all of the data will be made publicly available so more research can be performed. The TCGA will collect and examine tumor samples obtained from certain biorespiratory institutions treating patients with cancer. The project's four main components: a biospecimen core resource, 7 cancer genome characterization centers, and 3 genome sequencing centers, and a data coordinating center will collaborate in order to test methods and technologies, which will lead to the creation and organization of data that will be made open to the public for further research. While the TCGA research teams examine the DNA of 1,500 tumor samples of glioblastoma, lung cancer, and ovarian cancer, a list of target genes will be fit into each cancer type. Candidates will also be selected from specific categories of genes that are already linked to cancer. Gene categories include genes having abnormal structures or activity in a significant amount of tumor samples, well-known oncogenes, well-known tumor suppressors, and genes associated with known oncogenes and tumor suppressor genes by their amount of similarity or pathway association. The future of cancer research is full of many challenges that are both known and unknown. We only have a slight idea of where all of this work will lead. The findings that are occurring today can be compared to the expedition led by Lewis and Clark. Although they did not find exactly what they were looking for, their journey resulted in the collection of data that proved to be extremely valuable in many ways that were not expected beforehand. That is the end of my presentation, and here are a list of sources that I used for some of the information and pictures I included.